Good day, everyone. Today, our topic is about the respiratory system. So let's take a deep breath and let's begin. Remember respiration? We took it earlier. It is the release of energy from food inside living cells. All living cells, if they are alive, they are respiring. This is an important chemical reaction. Without none of us, none of any of the living organisms on this planet could have existed. It's a very important process. Now, respiration can be two ways. There are two types of respiration. They are either aerobic or anaerobic respiration. Aerobic means that it requires oxygen, while anaerobic means it does not require oxygen to take place. Fortunate for some organisms that have no oxygen around them. Examples of cells that respire aerobically: plant cells, animal cells, human cells, we respire aerobically. Well, we do have exceptions. Yes, there are some cells that respire both aerobically and anaerobically, such as yeast fungi. Yeast are fungi that are able to respire both aerobically and anaerobically. Human muscles actually do respire anaerobically as well when there is not enough oxygen, when the person is exercising vigorously and the lungs are not breathing fast enough and the heart is not beating fast enough. That is when the human muscles respire anaerobically. Now, I would like you to remember one thing. Releasing energy is what we use when we talk about energy being given out of the broken down food. We don't say food produces energy. It's a scientific thing. If you remember, if you have studied physics earlier, we cannot make, create, or destroy energy. We can just change it from one form to another. So we prefer you using the word releasing energy rather than using producing energy. Remember that. Now, cells release energy from sugars, for example, by aerobic respiration. And we're going to talk about one of the most important sugars that our cells use for respiration. It's glucose. This is the chemical reaction that actually happens in our cells. Glucose, with the help of oxygen, they react together, and the molecule of glucose is broken down to give off carbon dioxide and water. A lot of energy is given off as well. Please remember that we need to memorize this equation. For core biology, you need to memorize the word equation only, which is fortunate, isn't it? Now, moving on, in our muscles, during vigorous exercise, the muscles require a lot of energy. The person is running fast, for example, or exercising very quickly, and a lot of energy is required, but there is not enough oxygen. So the muscles do what? They respire anaerobically. Glucose is broken down, and a molecule called lactic acid, actually two molecules of lactic acid, are produced, and this produces a little bit of energy, which helps. But if there is a lot of lactic acid, a muscle can start to cramp. So we should warm up before going into any exercise to prepare our respiratory system and circulatory system so that more oxygen is delivered to the muscles and prevent any accumulation of lactic acid in our muscles. Lastly, in yeast cells, fermentation takes place in the cells. This produces energy from, or releases energy, remember, releases energy from sugars by anaerobic respiration. This reaction is actually useful when making bread and when making alcoholic beverages. Glucose is broken down into carbon dioxide. Carbon dioxide is useful to help to make bread dough rise, and ethanol as well is produced, which is made use of in alcoholic drink, such as beers, making alcoholic drinks. Finally, I would like to stress that, again, energy that is released from anaerobic respiration is much less than that produced or released by aerobic respiration. That is because glucose molecules are not completely broken down in anaerobic respiration. Remember alcohol that is produced by fermentation by yeast, it could be used as a fuel. So there's a lot of energy still in that molecule, same as in lactic acid. So just a note to remember that energy released by aerobic respiration is much more than that released by anaerobic respiration. So either way, aerobic or anaerobic respiration releases energy. Okay, so what do we use the energy for? What do cells need this energy for? Well, for one thing, our muscles need this energy for contraction. Organisms that have to maintain their body temperature, such as mammals and birds, more food is broken down in winter to supply the body with enough energy and enough heat so that the temperature stays constant in such warm-blooded animals, building new large molecules for cell growth and to help nerve impulses to travel through our nerves and to allow active transport. If you remember, this is a way by which molecules are moved from their low concentration area to their high concentration area against their concentration gradient. This requires energy as well. So that's why we do need that energy. Now, if I want to find out if respiration takes place in a sample, let's say I have a sample of soil. Maybe we got that soil from somewhere where we don't know if there are living organisms or not. How would you tell if the sample of soil, here we have one of the rovers that scientists sent to Mars, and samples of soil are repeatedly taken from different parts and analyzed to find out if there is life on Mars or other places such as the moon, or for example, or not. What would we look for? What should we search for? One thing you might think about is, let's view the sample under the microscope and look for cells. Yes, you're right. If we find small cells that we could not have seen larger organisms, we did not see any larger organisms, so we need a microscope. Yes, you're right. But what if we cannot see anything? How could we tell? Maybe the organisms are too small for our microscopes. If it's respiring, it's living. And if it respires, it should produce one of two things. Carbon dioxide? Yes, maybe we should test the soil if the soil produces carbon dioxide or not. Or heat. Every time respiration happens, heat is given off. That's why an organism has been in a living organism such as your brother or sister has been sitting on this chair earlier, because when you sat down you felt the heat on that chair, didn't you? So all organisms give off heat when they respire. If you want to test if a sample produces carbon dioxide, what would you use? If you studied chemistry, you might have come across lime water, a solution that is clear, but when you bubble carbon dioxide through it, it starts to become turbid. The longer, the more carbon dioxide that is bubbled through the lime water, the more turbid it becomes. It becomes cloudy or milky. You might want to describe it as that. We don't say a white color. We say either it becomes cloudy or milky or turbid. So this is a way by which you can find if carbon dioxide is in a sample of air, such as the air that you breathe out, or not. Here I have an experiment where a mouse is inside a container, and the container is connected to two other containers. Air is drawn out from the second container, the one on the right, and air passes through a previous container before going to the mouse. Lime water is found on the right container, and substance X is found on the left one. So lime water, we just said, what is it for? It's to test if air that came out of the container where the mouse is contains carbon dioxide or not. So we should expect after a couple of minutes maybe that the lime water changes and becomes cloudy or milky. But what about substance X? Why is it there? What if it's important? Is it important? Well, some could argue that the lime water that is on the right side changed to milky because actually air contains carbon dioxide. Well, they would be right. So I would have to prove that the air which came through the second container that contained lime water changed because of the carbon dioxide released by the mouse. How would we prove that? So maybe we should have added in the previous container something that would have reacted with the carbon dioxide coming in from the air around us. So maybe it should be a chemical that absorbs carbon dioxide. Wouldn't that be clever? Yes, so here in such an experiment, we're sure if we have a substance that absorbs carbon dioxide at the beginning, that the color change or the appearance that the lime water became cloudy or milky was only because of the carbon dioxide given off by the mouse. Well, let's move on. To test if a sample is producing heat. Now, here we need a container that will not allow heat to escape. This is actually, we have here two flasks where they are vacuum flasks where there is an area where there is vacuum between the two layers of metal. That this is a very good way to keep heat trapped inside and not escape, so we can record the temperature inside each flask. While in both flasks we have peas, seeds, and they have been germinating. What do we mean by germinating? Do you know what germinating means? It means that the cells have become active. They are not dormant anymore. Seeds, if you remember, are dry cells. They are not active unless they are soaked in water. So to allow the seeds to germinate, we have to give them some time in water, maybe a couple of hours a day, for example, in which seeds are soaked in water so that the water enters the seeds, they become active, chemical reactions start to happen, and as well respiration. Before that, no respiration would have happened. Now, you would notice here that the seeds were washed in a disinfectant. A disinfectant is a chemical that kills microorganisms. Well, yes, because microorganisms would have been respiring as well. They're living, okay? Bacteria, fungi, they have food around them. There is water in the cells, in the pea cells, so and the temperature is suitable, so they will grow and respire. So, how would we tell if the respiration was due to microorganisms or due to peas? We couldn't have told which is which. So we need to wash the seeds in a disinfectant to remove any microorganisms on their surface, as well as the container from the inside. It would be a good thing to do as well. Okay, so what also we have here in container B, in flask B, we have our peas boiled. Why would you do that? Boiling seeds would do what? It would kill the seeds. It would destroy the seeds. The enzymes would denature. The chemical reactions would have stopped. So why do we do such an experiment? Why do we repeat it with dead seeds? Well, maybe you want to prove that the change that happens in A was due to the respiring seeds and not due to an error. So actually, container B is a control experiment. It is done to prove that the changes that you have seen in container A are actually correct. Your conclusion is correct, okay, because you compared it with dead seeds that did not respire, obviously. They were also washed in a disinfectant. All right, so we have at the bottom a moist cotton wool. Why is it there? Moist, you might say, to prevent the seeds from drying out. Yes, you're right. And cotton wool, why not just a cap, plastic cap, to allow gases to enter and exit? The seeds are respiring aerobically. It requires oxygen and needs to get rid of carbon dioxide. So this will allow gases to move in and out, but will trap the heat. These two containers will be left for 5 days, in which the temperature should be monitored or taken every hour. Fortunately, here we have electronic thermometers that are connected to a computer, to a data logger. You will program such a computer so that readings are taken every hour for 5 consecutive days, which is easier than doing that yourself, isn't it? So let's assume that the temperature that we started with was 20 degrees Celsius, room temperature. What would you expect to happen in flasks A and B? Let's think about flask B to begin with. This has no respiring cells and no respiration is happening. Heat should not escape out of this vessel, so the temperature, if it starts at 20, should remain at 20, if this is a good vessel. Yeah? All right. And what would happen in A? I assume it should go up a bit. Yes, the temperature should rise a bit. We're just predicting, so we don't know. We have to do this experiment to find the temperature exactly, how much it rises. So this is what we should see in flasks A and flask B. What if we want to measure the rate of respiration in living organisms, such as germinating seeds or small invertebrates? Here we have an apparatus that we call a respirometer. It measures the rate of respiration in plants, small seeds, for example, or small animals, such as maggots or uh, insects or whatever. Okay, so the container could be as small as a test tube or maybe larger, but it has to be fixed with an airtight bung to a small capillary tube in which a drop of liquid, such as this red drop of ink, is inserted. The seeds will respire, and as we have learned before, respiration will take in oxygen and give off carbon dioxide. Well, if oxygen is removed from the air inside the container and carbon dioxide is released in its place, nothing will happen. The amount of air inside the tube is still the same. So over the left side, we have here small pieces of soda lime. This is a chemical that absorbs carbon dioxide. So now we have something different. Oxygen is taken in by the seeds, and carbon dioxide that is given off is also absorbed by the soda lime. This, in conclusion, causes that amount of gases, oxygen and carbon dioxide in the tube is less, it decreases, and therefore the pressure inside the tube is less, causing the pressure outside to push our red ink drop in the capillary tube towards the seeds. So now we can measure, if we have a stopwatch with us, here we can see in the diagram that our drop has moved 4 centimeters in 4 minutes. So the rate of respiration of these seeds is 1 centimeter per minute. We can use this respirometer to examine or investigate the changes in temperature. Do they affect the rate of respiration? We can compare the rate of respiration of different seeds, for example, maybe at different seasons, whatever. So this is a way to tell how the rate of respiration changes with changing any other factor. So this causes the red liquid to move to the left, and we can calculate the rate as we have did in a moment before. Now, our respiratory system. Yep, let's go to that. We have lungs, very efficient organs that help us to exchange oxygen and carbon dioxide to oxygenate the blood. here I want you to note that drawings of the human body is always a drawing of a person standing in front of you. So this is actually the left lung and the other one is the right lung. The heart is nestled between the two lungs over here. And then let's start from the very top where the larynx is. Here is where our voice is made when air enters or exits the lungs. The trachea is the tube that carries air down, and then it branches into two tubes, smaller tubes that we call a bronchus, each bronchus. Both are called bronchi. Bronchi again start to branch off into smaller tubes that we call bronchioles, and at the very end of each bronchiole is a group of air sacs that we call alveoli, or if we refer to one of them, we say it's an alveolus. The ribs help to protect our lungs and the heart, and they are moved around by intercostal muscles. We have also a sheet of muscle at the bottom we call the diaphragm. How does gas exchange happen? Well, how are alveoli actually adapted to allow gas exchange? How does that happen? Well, there are many things to consider, one of which the wall of the alveoli is very thin. It is actually one layer of cells thick. So that's a very small distance for molecules to pass through, which is very good. This will speed up diffusion. So oxygen can diffuse very quickly from the alveoli into the capillaries around them. So the oxygen is moved by the blood to the, or delivered to the cells that need oxygen for respiration, and carbon dioxide will diffuse the other way around from the blood to the alveoli. One other thing is that there's a thin layer of moisture inside alveoli which will help to keep the cells from drying out, because it is one sheet of cells and air is rushing in and out all the time. These cells might dry out. They don't, fortunately, because of this layer of film of water. And it allows the gases to diffuse. They dissolve first inside this layer of water and then diffuse from cell to cell until they reach our blood cells. So this helps to speed up the diffusion. Also, we have many of these alveoli. They are numerous. And what fascinating is that the size of both our lungs is only roughly the size of basketball, while the surface area, if we counted all the alveoli and opened them up and flattened them down and counted or found the total surface area that they provide for us, they give us a surface area of a tennis court. Amazing, isn't it? Yes, we have a very efficient organ that we should take care, not to destroy. So this is a very good surface area that will allow us to become active organisms. We can run, we can climb, we can jump, we can do whatever because of this efficient organ. One of the famous questions that comes along this chapter is how our alveoli or exchange surfaces are adapted to increase gas exchange. We have mentioned them in the previous slide. Let's revise them. The alveoli have very thin walls. This decreases the distance for diffusion, and this increases the rate of diffusion of gases, such as oxygen and carbon dioxide. Alveoli have moist walls, which allows gases to diffuse in this water and diffuse much faster through our cells. Alveoli are numerous. This increases the surface area, and therefore the rate of diffusion is much faster. Now, what if the question was not that specific? What if it said, how are lungs adapted to increase gas exchange? Well, you could mention all these 3 points, and you could add to them as well that the alveoli are surrounded by numerous capillaries, and these numerous capillaries allow oxygen to move out of the lungs or out of the alveoli much faster, and they help to keep the concentration gradient high. So there is always a high concentration of oxygen inside the inside the alveoli, and a very low concentration of oxygen in the capillaries. So oxygen diffuses faster from the very high concentration to the very low concentration, which helps things to move faster, oxygen to diffuse faster. So this is a way of, you have to read the question carefully to make sure if it's requiring you to answer how the lungs are adapted or how the alveoli are adapted to increase gas exchange. So, does air differ when we breathe? If we compare the air that we breathe in with the air that we breathe out, yes, there will be a difference. Oxygen in inhaled air is around 20%. It goes down to 18 or 16%. That's because, obviously, we have been talking about this for too long already. Oxygen is carried away by our red blood cells. Carbon dioxide, on the other hand, increases from 0.04% to 4%. That's because this is the major way by which we get rid of carbon dioxide. This is released by our respiring cells, and oh, do we have many cells that are respiring, don't we? Nitrogen stays roughly the same. It is not used by our cells. The temperature is variable according to the temperature of the air that we inhaled, according to the temperature at which we are sitting in, but usually it's higher when we exhale. That is because the air is warmed by the body tissues. Humidity also is variable, but it becomes greater. We did this as children, don't we? We still do it as adults, do we? So when you breathe out on a cold day, you find that the air has a lot of moisture, and you can see actually the condensation, like dragon breath, do we call it? Did we call it as children? What did you call it? So air is moistened because it has been moving through moist body cells, such as the alveoli, for example, and they carry this extra humidity out. If we compare inhaled and exhaled air, we will use this brilliant apparatus. It could be 2 test tubes or 2 conical flasks. Either way, they should be very tightly secured to a group of tubes. You would benefit from memorizing this diagram. So let's look at it carefully. We have a mouthpiece in the middle through which the person doing this experiment will breathe in and out. Out of this mouthpiece, 2 tubes come out. One of them on the right side is a bit short and it doesn't touch the lime water inside the tube. The other one is long enough to be inside or submerged into this lime water. In the other tube, if we're looking at tube A, if we have a long one, then the other one should be shorter and not touching the liquid or the lime water. And if we're looking at tube B, we have a short one, so there should be another one long so that it is submerged into the lime water. So you should memorize that each tube, A and B, should have one long tube and one short tube, and the mouthpiece is connected to 2 tubes, one of which is long and the other is short. This arrangement will allow the air, when you breathe in, so air is coming out of your mouth, it will move to the left, enter the long tube in A, and it will bubble off and it will come out of the tube. So actually A is the tube that will help to find if exhaled air contains carbon dioxide or not. So A here shows exhaled air. On the other hand, tube B, when we breathe in, the air will move from the air outside the tube through this long tube in tube B, bubbling out of the liquid into the short tube and into the mouth of the person breathing in. So tube B is for inhaled and exhaled air. What change would you expect? So if B is for inhaled air and A is for exhaled air, and we have just said that exhaled air contains much more carbon dioxide than I expect, yes, A would become milky first. B will not change milky, not even, it will take a bit more longer for B to change color because there is a very small amount of carbon dioxide in the air around us. We have just said 0.04%. To measure the lung volume, we have this simple apparatus over here. We have a big container in which another container is placed. The container is filled with water, and then a breathing tube is inserted so that a person is asked to take a deep breath and then fully breathe out as much as he can to make sure that he breathes out fully. So the air that he breathes out displaces the water inside this jar, this bell jar, and we can use the numbers, the graduation, to tell how much air has been breathed out. In this diagram, we can see that 2.5 liters of air was breathed out. We can repeat this experiment by removing this rubber bung, pushing the bell jar deeper into the liquid so that all the air leaves the bell jar, and replacing the rubber bung and repeating this more than once to get reliable results. Yep, good. The effect of exercise on breathing rate. One of the very common questions we are asked every year to design an experiment. Here you might want to practice it yourself, carry out this experiment to show how exercise affects the rate of breathing. What exercise would you choose? There are many exercises to choose from, whichever you choose, okay? How are you going to make your experiment fair? In every experiment, I would like to step back a moment to remind you, in every experiment we have an independent variable. This is the variable that you change intendedly. You want to see the effect of that factor. Okay, so here our factor is exercise. We would like to see before exercise what happens. So maybe you would want to find the breathing rate before exercise and then after or during exercise, whichever you want. And how would you want to make that experiment fair? Obviously, you have to keep some other factors constant. So if you're exercising, you should be always doing the same exercise. One important thing. Maybe you should be using the same running shoes. Maybe you should be exercising at the same room temperature. Could be. On the same surface. Maybe if you're running, you want a smooth surface or a rough surface, whichever. It should be the same. So factors that should be kept the same. And one of the most important factors here, if the chosen exercise was running, you should think about keeping the speed of running the same and the distance the same as well. Maybe the time as well should be kept the same. So maybe you want to control that by using a treadmill, for example. Right? So these are things that you should keep the same to make your experiment fair. What should you do to make your experiment reliable? So maybe you want to make more than one reading. Maybe you want to have more than one individual taking this experiment or involved in taking part in that experiment. So maybe you have to have many volunteers. But of course, if you have many people, they should be of the same age, same gender maybe, same fitness. This is important as well in such an experiment. So what would you do to make sure your experiment is accurate? Now, accuracy has to do with measuring and using a better way to measure or a better tool to measure. If you were going to just look at the person and count the number of times he's breathing in or out, maybe you will miscount. So you might think of a way to make sure you don't miscount. Maybe you want to take a video of him and recount and make sure that you have the right count. Or maybe you want to use a device that will be measuring maybe how many times the chest has stretched out and maybe. So this is a way to measure. This is a device that uh, measures how many times the person breathes in. So the wires stretch and then return back. So this is a way to make your experiment more accurate. And maybe you want then to plot your results on a graph so that people can decide if you have correct results or not and compare. Now, to look at breathing rate graphs, here we have a graph where the lung volume is recorded. A person has been taking several breaths and then asked to take a deep breath in at the second second number 2. He took a deep breath in, so the volume increased greatly in his lungs, and then it dropped down at second 4, and so forth. In such a graph, you might be asked to calculate the breathing rate. So let's take the first 2 seconds as an example of his breathing pattern. He's taking a breath and then another breath for in the first second. Again, 2 more in the second. So in the first 2 seconds, this person took 4 breaths. So if 4 breaths in 2 seconds, so how many breaths would he take in 60 seconds, which is a minute? Well, cross multiplying that would get 120 breaths per minute. That's a bit too much. That's a person who's exercising actually. Could be. Normally, a person would breathe about 15 to 18 times a minute. So this person is breathing quickly. It could be a child. Then another question could be, use this graph to tell the maximum lung capacity. So the lung volume or the lung capacity is how much air your lung can take. So the person, when asked to take a deep breath, he took a deep breath, and how much air did he take into his lungs? Yes, the highest point over here was between 6,000 and 7,000. So let's say estimatingly 6,500 milliliters of air. So this is the maximum lung capacity. How much more air was breathed in when the person took a deep breath? Here we have several steps. We have to first determine when he was not taking a deep breath, he took about 4,200 maybe milliliters of air. And when he took a deep breath, he took 6,500 as calculated earlier. So the difference between that would give us the answer, 2,300 milliliters. So we have finished our topic. I hope you have understood, you found it easy to understand, and we'll meet in the next chapter. Have a lovely day.